everyone, good evening, welcome to the class. Hi everyone, good evening. I hope that all of you are doing great and I hope that you can see me and can hear me clearly. How are my achievers today? Good evening, yes, oh, awesome. Ranjit sir, if it's possible, can we have the chat? Thank you. Yes, everyone. So how are you all? Do tell me, how are you all? How is the energy? How has been like whole week from Monday till Thursday? Yeah, still just one more day. Yeah, and of course we are here. Good evening, Nikita. Good evening, Rashmi, Rafiq, Mona Lisa. I'm good. How are you? Epic, Shubhi. You're perfectly audible and visible. Thank you so much. Good evening, Rashmi. I'm good. How are you? Thank you for asking. So awesome, everyone, to see you, all of you here. Good evening, Prachi. Good evening, Soham. Good evening, Bhumika. Hi. Yes. So, as you can see from the title of our today's session, we will be doing some high order thinking skill questions, right? Now, this is very interesting. Yes. Good evening. Good evening to Akansha, Hema, and Kalavati. Good evening. So, we are almost like we actually finished our first chapter, that is the fundamental unit of life, right? And especially in the class 9. And we'll be starting very soon about the second chapter, so stay tuned for that also. When we talk about the first chapter, that is fundamental unit of life, it's a very easy chapter and it has very interesting questions. But you know what, I thought that many of you are asking about, ma'am, some, some high order thinking, like basically some tough questions so that we can test ourselves. And of course, these questions will be helping us to actually look for some other important concepts which are not there in CBSE, but of course that if we know about these concepts, we'll be able to clear, these big and tough questions. And these, of course, these questions, usually asked in the NTSC examination or in other competitive examination like Olympiad also. So I feel that all of you are ready, and so this was a need, right? That's why we are here with this amazing session. Epic energy, yes, yeah. Mohammed, I will try my best, but yeah, I will not be able to speak in Hindi, and of course all of it. We, the majority of the students over here in our class, understands English, so we'll stick to that. But thank you for letting me this know. I'll try. Yes, good evening, Sanjeev, Venkat, Salma, Nikita. Awesome. Anjali, Balram, everyone quickly give me a thumbs up and we'll start our session, right? Epic, what's your name? Yes, awesome. So we are clear with this, right? Let me quickly take you through with this, right? We have Byju's mini learning program, right? And of course, you can easily avail it by using the code which is YT free. And of course, this particular classes, right, that we have are really very interesting. And what you can expect in these classes is something that I think all of the students loves to have. 2 teacher models is there. So yeah, you'll be learning with 2 teachers. There will be a personal mentor to help you and guide you. You have live interactive classes. You'll be having quizzes and the test. And apart from that, yes, you can actually pick the timing of your own classes. So many of you have already tried, and I would request all of you to also try it, right? It's absolutely free. If that's the important thing. So you try it out. You will have 3 sessions, and I'm sure this will be able to give you understanding about how the classes are. So of course, please do try it. And of course, this is there only for the limited period of time. So please try it out. And of course, we have a Telegram group. I don't have to tell you much about it. You are already there. And if you are not there, join the Telegram group. The link of the, the group is there in the description box below. Sorry, excuse me. Yes. Yes, Adil. Yes. Aditya. Ahir. Nice. Purnima. Tablet will not be, no, you can actually try with it. You will not be needing the tablet for this as of now. Yes. Awesome, everyone. So let's get started. My name is Ankita and welcome, everyone. Hi, Safan. Welcome to the class. And yes, good evening, Mahalakshmi. So everyone, now I'm starting the class. I would request all of you to focus over here, right? So in today's class, those of you who have just joined now in today's class, we will be discussing about the questions which are high order thinking skill questions. Basically, these are the tough questions that requires us to apply the applications of it. Whatever we have studied, these will be little more of application level questions, right? And we will try to solve them out. My name is Ankita and you are on Byju's 9th and 10th channel. Welcome to the class. So let's get started over here with the first question. Yes, here we go, everyone. Now, I want all of you to be focused over, please focus over here. We don't have Menti for these questions because these are a little bit tough questions, but we will be having the Menti quiz for the other questions also. So, yeah. Yes. Okay, so let's get started, everyone. I want you to write the answer in the comment section below. A person takes up the concentrated solution of salt. Interesting, right? For example, if I'm a person, if I'm drinking the concentrated solution of salt, just mixed in water, after some time, we starts to vomit, right? What is the phenomena responsible for such situation? Explain. Very good, Monica. Very good. No, Chitra, you're right on time. We're just starting with the very first question. All of these are hot questions that we have from Fundamental Unit of Life. Everyone, start answering. I want to see. I hope that you're revising right, and that's very good. I can see the answers. Very good, Mahalakshmi, Rafiq, Ashwarya. Hi, Prachi, Chitra, Monalisha. It's okay, Sarah. Welcome to the class. Very good, Adil. Soham, Mukesh, Venkat, Rafiq. Very good, Anjali. Salma, hypertonic solution. You guys are super awesome. Bollywood updates. Oh, interesting name, but what's your real name? Purnima. Vanshita, Cutie. Yes, you will be able to get the classes, right? And you can try those classes and then, of course, you can take a decision along with your parents. Yes, Dipanshu. Very good. Firdaus. So, let's quickly, let's just look at the answer, right? We have the cell and, of course, if we add it in the hypertonic solution, what will happen? Of course, the water will be moving out from it, right? It will be losing the water. See what is happening over here. So, if we drink water, right, of course, what is happening? The digestive tract that we have, right, and it has the cells. What will happen? The salt water that is going, what it will do? The salt, right? Of course, as there's a concentration difference, what will happen? The water from these cells will be coming out and the cell will shrink. And as there's water, we'll see the vomiting happening. Are we clear? Yes, are we clear, everyone? And of course, there will be loss of water and of course, the dehydration as the cells are losing the water. Our cells in a dehydrated state and of course, that will be causing the vomiting. Are we clear, everyone? It's a very easy concept, right? Now, if you think of, the question looks really very tricky, but of course, the understanding and the important, if you are clear with the concept, it's really very easy. Over here, super proud of my students, my bachas. I'm super proud of you. And over here, this is the way we can write the answer in the examination if this question comes. So, concentrated salt solution is a hypertonic solution. So, causes irritation and excess dehydration in the walls of the alimentary canal due to the exoosmosis. So, water is moving out. So, exoosmosis is happening. And because of that, of course, what will happen? There will be uncomfortable stretching of the digestive tract. And of course, because of that, we will see the reverse movement and hence the vomiting. Okay. Yes. Are we clear with this? Very good. So, if you want to take a screenshot of this, you can, right? And of course, we'll move ahead to the next question. Here we go to the next question. So, Adil, quickly for all of you who just want to quickly revise this, all right? So, of course, as we drink, right, the salt water, it's a hypertonic solution. As it goes inside the digestive tract or the alimentary canal, what will happen? The excessive water will move out. Basically, the water which is there in the cells will move out and the cells will shrink, right? Of course, there will be dehydration in the cells and that causes the vomiting. That's it. Okay, question number 2, everyone on your screen. What will happen if the chloroplast is taken out of the cell and illuminated? Oh, interesting question, right? For 2 marks, everyone, quickly tell me. Yes, what will happen? Very good, Zaid. Of course, that's a very common point that, plants will not be able to perform photosynthesis. Yes. No photosynthesis, hence no food. Very good, Nilima. Very good, very good. Please, everyone, please answer. Yes, Monica, no photosynthesis. Yes, plants will not be able to perform photosynthesis. Very good, Soham, Sudhanshu, Rafiq, Nikita, Prachi, Shilpa, Venkat. Colors of the leaves. Yes, colors of the leaf will not be green. Very interesting. So finishing, plant will not be able to gain the energy and due to the loss of photosynthesis. Yes, yes, makes sense. Mahalakshmi, very good. Yes, Balram. Interesting, very good. So of course, when we talk about, we know that chloroplast plays a very important role, and it is there only in the plant cells, and of course, chloroplast plays a very important role in process of photosynthesis. So of course, it will be absorbing the light, right? And of course, and hence that's how the process will occur. But if it's not there in the plant cells, and even if we take it out and illuminate it, what will happen? Nothing will happen. It will not be able to make, right? What do you think? Very good, very good. So if we have to frame the answer, we can frame our answer in such a way that chloroplast is a semi-autonomous organ. That means that it's a very important organelle, right? And of course, it has its own DNA. On removing the chloroplast from the plant cells, the cell of course will not be able to survive. Chloroplast is a site of photosynthesis and of course glucose production, and on removing it, plant will be deprived of the food and will not be able to survive. Right? Are we clear? Yes, junior word. What's your name? You have to tell me your name. No life is possible. Very good. Yes. Very good, Nilima. It's called the kitchen of the cell. Absolutely correct. Eliza, good evening. Welcome to the class. Yes. Semi-autonomous means that basically they can make their own proteins, right? Right, and they have their own DNA. They're kind of independent organelles. Sorry, not the organs, organelles. Hi, Gaur. The color answer is also correct. Yes. We can say that, of course, it will not be, of course, the green color that we have. It's not there. The leaves will not be green. But yeah, that's a very far-fetched, because of course it will not be the primary answer, but it can be the third point. Awesome. So I think we are clear with this question, right, everyone? Come on, come on, come on. A very quick thumbs up in the chat box. See, come on. We should have the active interaction so that we all are enjoying a class. And please don't forget to hit the like button for the video. As you're showing a thumbs up, hit the like button for the video. Is it Gaur? I will be. We usually upload. Just check again. Awesome. Let's move to the question number 3. What are centriole? And we have to write about their function. A very easy question. Write a very easy question. I'm sure you remember about the centrioles, though we haven't discussed in detail in the cell organelles, but we had a discussion about these when we discussed about the cell division, right? So, we are talking about the centrioles. Now, centrioles, I'll tell you, are really very important, right? And of course, when we look at the structure, they're the cylindrical structures. Yes, and they help in the cell division. Very good, Nilima. Yes. Very good, Salma. All the best for your test. Very good, Zaid. Very good, Nilima, Rafiq, Chitra, Soham, Venkat. Yes, Prachi helps in cell division. Very good. Yes, it helps in the cell division. And when we talk about its structure, these are the centrioles, right? And of course, they are present in the cell, and during the cell division, they'll move to the opposite pole. They will help in the formation, right? They'll release the spindle fiber. Basically, they help in the formation of spindle fibers. And these spindle fibers will attach to the chromosomes and will pull the chromosomes, right? So, they play a major role in the cell division, help in the formation of spindle fiber, and of course, help in the production of cilia and flagella in the prokaryotic organisms, where we have those cilia, and of course, in the eukaryotic organisms also. Flagella and cilia, I'm sure you are aware about, right? If we talk about the bacteria, they have a flagella which actually help in the movement. And of course, on the paramecium, we have these small finger-like projection. These are the cilia, again, which helps in the motion, right? So, are we clear with this? Very good, Sudhanshu. Very good. Bollywood updates. What's your real name? Only in animal cells. Very good, Soham. Pranjal, thank you so much. Very good, Aliza. Yes, I'll explain. So, cilia and flagella are the small locomotory finger-like projections. So flagella, of course, is a bigger one. Of course, the flagella, it's usually as a big tail, right? Of course, if you see in the bacteria, they'll have this. This is a flagella, right? This is the flagella when we talk about it. And of course, when we talk about the paramecium, they have these small finger-like projections, and these are the cilia. Okay, and both of these flagella and cilia helps in the locomotion of the organism. So flagella, of course, we can see in the bacteria, and cilia in the paramecium. So are we clear with it, everyone? Yes? Falak, yes. Oh, that's a nice name. Great. So let's see how we'll be writing this in the examination for 2 marks. Centrioles are the paired, very good, I think many of you are writing this, right? Centrioles are the paired barrel-shaped organelles located in the cytoplasm of the animal cells, right near the nuclear envelope. Centrioles plays a role in organizing the microtubules, right, of course, the whole structure, and they plays a very important role. And of course, they help in the spindle fiber formation during the cell division also. Very good. So again, I would request you to take a screenshot of it. You can take it later also, but we are clear with this. Now, I know that in the NTSC examination and in the Olympiad, we don't have subjective questions per se. But of course, we know that if our concepts are clear, we'll be able to pick the correct answer. So these questions are really very important from those competitive exams also. So please focus over here. Very good. I can see crystal clear. Yes, today you will be getting the notes of this particular chapter, right? This PDF you will be getting on the Telegram. Yes? Very good. Nice. So I think all of us are clear. Yes, Monica, I will be. Nice, Ashwarya. Very good. Great. Awesome. So let's see the question number 4. Why are plant cells more rigid than the animal cells? Oh, I can see the answers already. Because of the cell wall, vacuole. But what do you think is the reason behind it, right? What do you think would be the reason behind it? Why are plant cells more rigid than the animal cells? And of course, it's a 2 marks question. Cell wall, cell wall, cell wall. I can see a lot of cell wall. So let's quickly discuss it out. So over here, we have the plant cells over on that side, and this is the animal cell. So animal cells have big vacuole, as you can see, and of course, plant cells have big vacuole, and in animal cells we have small vacuoles. Nucleus, ER, mitochondria, cell membrane, plastids are still cell membrane. We have it both in the plant and the animal cells. Plastid and cell wall are specifically there in the plant cells. So the cell wall plays a very important role. We know that cell wall is made up of the cellulose. Very good, Rashmi. Very good. Draw and enjoy with joy. Yes. So, the cell wall plays a very important role, right? It's really very rigid, right? And because it is rigid, it provides a shape to the plant cells also. Now, when we talk about it, we know that plants are there outside, right? And they need that strength. Whereas in animal cells, right, they're kind of well protected. So of course, that's one of the reasons that, plant cells have the cell wall, and of course not in the animal cells. And let's see how we can frame our answer. Cell wall consists of plant cell consists of a cell wall, which is absent in the animal cell. This membrane is rigid when compared to the cell membrane, as it is made up of cellulose and is freely permeable. Cell wall is not only giving the shape to the cell, but also protect the cell from the mechanical damage and the infection. So this is important, right? So cell wall is really important. It's rigid, provide the protection also. It's freely permeable also, and of course, help in preventing the mechanical damage and the infection. So are we clear with this? Yes? Very good, Ashwarya. Very good, Rashmi. Very good, Chitra. Yes. Rafiq. Yes. Very good. That's very good, Nim. Niranjan, Venkat, Rafiq, Pranjal. Great. So I can see lots of thumbs up. Let me make a smile for all of us over here, and let's move ahead. We are moving to the question number 5, everyone. Let's quickly do this, right? Here. What would happen if gamete formation involved only mitosis? Now, can you see these questions? Now these questions are not very direct, right? They're kind of very tricky. So just imagine what would happen if the gametes, like sperm and the ovum, is formed only by mitosis. So we have two important cell division types, right? Yes, we have two different types of cell division. Now we have to find out only if mitosis is happening. Cutie pie, right? It will be great if you can tell me your name. So, of course, once you'll get the email and of all other procedure, there will be a team member who will be reaching out to you, right? And you can explore more on the website. Very good. So, just focus what will happen if the gamete formation occurs only mitosis. We know that in mitosis what happens? We know that in mitosis, the cells will be getting duplicated, right? And of course, we will have the two daughter cells formation. We'll see the identical copy, right? Now, we know that the sperm and ovum have how many chromosomes? 23, 23. Right? But of course, to over here to come 23, of course, it undergoes the mitosis process. So it will be diploid, right? And of course, that will not be a normal condition. So let's see what will happen. If only mitosis was involved in the gamete formation, we know that it will be 44xy, 44xx, and of course, it will be 88. So can you look at the numbers? The numbers are huge, right? Yes? Are we clear, everyone? So what will happen? Of course, the number of chromosomes will be doubled. So just you have to focus over here. We know that in the process of mitosis, what happens? We will see the duplication, right? We will see the identical copy formation. So when we talk about mitosis, of course, we'll see the duplication of it. But when we talk about the sperm formation or the ovum formation or the gametes formation, we know that it has two divisions, right? First, we have reductional division and then the copying. So, of course, if we use all our knowledge over here, we'll be able to see that, of course, the chromosome number will getting doubled. So, this is how you can write the answer. Each organs have a fixed number of chromosomes. If gametes produce instead of mitosis, each gamete would be diploid and not a haploid, right? So, what will happen after fertilization? Diploid gamete, the zygote would become 4n and not the 2n. And of course, with each of this, the number of chromosomes will be there, right? And this is why the cell organism will not be able to maintain the whole process which is happening. Yes. I hope this is clear, right? So I'm just moving aside. It's a very trickier question, everyone. It is very trickier question. It's okay, Nikita. You can watch the session after some time also. No worries. Great. Very good, Zed. Prophase is the longest phase. Yes, it is. Awesome, everyone. I'm super proud of you that you are clear with this question. Now, let's move to the next question. Question number 6. The cells of rat and elephants are same in size. What do you think? The cells that we have in you and me or in the rat or in the elephant, if we just say if they're same in size, do you agree with this? You have to justify your answer. What do you think? Do we have same cells? Or if you compare rat and an elephant, do you think they have same cells? If yes, then we have to justify our answer. I can see a lot of yes and a lot of no. What do you think? Yes? The elephant will have more cells, right? Because of the size of the animal. Whereas in compared to rat, rat has less number of cells. Think about it. See, I'm sure you must have seen a small baby, right? And of course, eventually with the time, there will be growth happening. And of course, that baby will develop into an adult, right? So what is happening over there? The cells, of course, are same, right? Of course, of the same type or different type. But what is happening? There's more cells increasing. The number of cells are getting more and more. And that's how the size of the organism will change. So, if we have a elephant and a rat, right? What we know that the size of the cell definitely is kind over here. Can you see the elephant? See? Look at the elephant, right? You can see the number of cells is more. Right? a number of, and they're just increasing. Can you see this over here? Look at the elephant, right? Okay. And of course, in the rat will have less number of cells, right? See over here, this is the elephant and this is the rat. So of course, the size of the cell will be same. But as the organism increases, right, if it has more number of cells, the size or the weight of the organism will increase, right? And if an organism with a very small cell, a less number of cells, of course, it will be like a rat. So this is how we can frame our answer. A very interesting question. Oh, you had this question in chapter class 8. Interesting, very interesting. So the size of the cell does not decide the size of an organism. If the number of cells that differs make the difference, so the number of the cells that we have in the organism will be the one will be deciding what will be the height of the organism, or size of the organisms, right? So because the size of the cell is different, does not vary in elephant, won't have a larger cells than rat, and it has greater number of cells as compared to the rats. That's why the size of elephant is more as compared to the rats. Easy? Very good. So the one thing that we have to remember is that number of cells will be important, not the size of the cell. Right, are we clear, everyone? Yes, a quick thumbs up. So what is important is the number of the cells. Right? More the number of cells, more huge organism will have. Less number of cells, of course, we will have a tiny organism. Clear? Oh, nice. Lots of thumbs up. Very good, very good. Let's move ahead to the question number 7 over here. What is the relation between a chromatin material and chromosome? 2 marks. 2 marks question. What is the relation between a chromatin and the chromosome? Interesting question, right? Both of them, of course, have the important part. Yes, quickly tell me, everyone. It's the easiest question for 2 marks. Right? Over here, we're talking about the chromosome and chromatin material. So this is chromosome, right? And what is chromatin? A very dense form that will be making the chromosome is the chromatin. Then we have the DNA. Right? So I'm sure all of you are aware about this. Very good, very good. So let's see over here. This is the chromosome, and if you open the chromosome, what we'll have? We'll have the chromatin, right? And if you open further, we'll have the DNA. So when we talk about the chromosome, we know that it is a highly coiled structure that forms this particular shape. Now the chromatin will have what? It is a dense structure, right? And over here, the DNA is wrapped on the proteins, right? These are the histone proteins. Which proteins? Histone proteins. So DNA will wrap onto these, right? So of course we have the chromosome and this is the chromatin. Let's take a closer look over here. This is what the chromatin looks, right? So here we have the condensed form, right? If we open this chromosomes a little bit more and more further, we'll be able to see the chromatin. So are we clear? The relation that they have is of course both of them plays a very important role. The structure is different and of course their composition, right? And how they are formed is different. The composition means of course they have the DNA and the proteins, right? But of course chromosomes is visible with the help of a microscope during the cell division, right? Only during the cell division we'll be able to see the chromosomes. Very good. So over here you can frame your answer. The DNA is packed by a special protein called as histones for chromatin that actually forms the chromatins, right? And chromatin fibers will further condense to form the chromosome, right? And of course DNA packaging, whereas of course chromosomes are the higher order of the DNA packaging. So in terms of packaging material, chromosomes are over here and the condensed, chromatin is a little bit lower over there. Yes, there is no Menti today, Bhuvana. Yes, we don't have Menti today. Renochromatids are this. So the one part of the chromosome that we have is the chromatid, And we have basically two part, right? And these are called sister chromatid, and they will come together and they will form a chromosome. So these are the chromatids. Are we clear, Renu? Renu, are we clear? And all of you, are you clear? Great. Quickly give me a thumbs up, everyone. We'll move ahead. I hope that you're having a lot of fun in the class. I know that question and answers are a little bit, not so great to learn, but you know that this is important from the exam point of view. Yes? Very good, Renu. Very good, Shashi. Good evening. Welcome. Awesome. Love to see your excitement and the enthusiasm. Yay, I'm super proud of you. I understand, Chitra. Yes. Here we have question number 8. What would happen to the life of a cell if there was no Golgi apparatus? Now, I'll not be discussing this answer. Come on. You know the importance of Golgi apparatus in the cell, right? What happens? I can see one answer. No lysosomes. Nilima, very good. No transfer of proteins. Bhuvana. Shashi, thank you so much. Nilima. Neelam is saying no lysosomes. Hari, transport will stop. Ashwarya, lysosome will not be formed. Venkat, no transportation. Prachi, no packaging. Rashmi, cell packaging will not happen. Very good. Transportation and modification. Nitika, no lysosomes. Ami, Deshmukh, Monica, Sara, Soham. History. Very good. Basically, Chitra is saying no Golgi apparatus, no life. Kind of true, right? Very good, Rafiq, Sunita. Yes, so let's quickly take a look, everyone over here. We know that Golgi apparatus is really very important, and they plays a very important role in the cell functioning. They are the flat membrane bound organelles, right? They have cisterns over here like this, right? And of course, they're stacked parallelly. And they are responsible for the packaging, transportation, and for the modification of the material that are synthesized near the endoplasmic reticulum, like lipids and proteins. So if Golgi apparatus are not there, nothing will happen, right? Of course, there's no lysosome formation, no formation of plasma membrane, right? And of course, the cells, organelles are just waiting. Oh, they'll not function. They'll be like, oh, I will wait. My protein is on the way. I don't know where it is, right? So, of course, the cell functioning will get hampered. Bhuvana. So, cisterns over here, can you see this particular structure that we have over here? These are one, and of course, they are stacked parallelly to each other. Very good, Lakshmi. You're joining the class after a long time. Ruqayya. But your name is written over there. That's why I was calling you out. Yes. Great. I hope that all of you are clear. Yes, everyone. So over here, this is how we can write the answer. Very good, Monica. I can see your thumbs up. Crystal clear. Very good, Rashmi. Membrane genesis will get hampered. Yes, Ruqayya. Now I know that. Very good, Salma. Prachi. Absolutely correct. So over here, we have this answer. It's for 3 marks. So just frame your answer in such a way that you're covering all the important points, right? So let me just move aside. Take a screenshot, everyone. Right? Take a screenshot. And this is how you can frame your answer. Very good. Moving to the next question. Question number 9 we have. The plasma membrane is permeable to water. How does a cell show endoosmosis or exoosmosis? Interesting. First of all, tell me, what is osmosis? Yes, what is osmosis? Right? What is osmosis? Osmosis is nothing but a simple process. Of course, we'll see the movement of the water molecules from the high concentration to the low concentration through a semi-permeable membrane. Yes? It's okay, Lakshmi, got it. Yes, very good, Mahalakshmi. Yes, Zed, Arshia, Chitra, Neelam, Ruqayya, Shweta, Balram. Very good, yes. Bhuvana, yes, Soham, Renu, Sunita, yes. So we are clear what is osmosis. Exoosmosis means water will, endo. Remember everyone, whenever we have the word endo, it means in. And when we have exo means out. So of course, water is moving in its hypotonic solution, when water is moving out is hypertonic solution. So in endoosmosis, water will move inside, and in exoosmosis, water will move outside. The plasma membrane is freely permeable to water. How does the cell show this, right? We know that. Of course, we'll have the water movements, and because of that, of course, we will be able to see the change in the shape of the cell. In hypertonic solution, the cell will shrink, right? Exoosmosis will occur, and in the hypotonic solution, endoosmosis will occur, and this is how we'll see these cells changing its shape, right? Hippo, remember that? Hypo, hippo, hyper. Yes. So this is how we can frame our answer. The movement of water into and outside the cell depends on the concentration of solute that is present in the water, right? The cell has a lower salt concentration than the water present outside the cell. The water will move from cell to the outside, causing the exoosmosis, and the opposite will occur in the endoosmosis. So over here we have the definition of it, everyone. Quickly take a screenshot of it, it's important. Yes, I hope that you have taken a screenshot, right? Very good. So that's how we'll be able to write this answer in the examination. Yes? So see, it's very interesting. So we are clear with this. Now let's move to the question number 10. You know, we have 5 more questions after this. So let's quickly finish our class, everyone. Viruses have genetic material as well as the protein, but still are not classified as living. Explain. Now I'll just wait for you all to answer this question. It's a very interesting question, right? We're talking about the virus. Now, virus is living or non-living? We have discussed about it, right? We usually call virus as a non-living. It is living only inside the body of the host. It's non-living outside the body of the host, right? It uses the host body mechanism for the process of reproduction or for the process of replication. So we are clear that viruses have genetic material, they have proteins also, but still they are classified as living or non-living. So we are clear with that, right? So here, let's see, we have to explain this. So viruses have this very amazing structure, right? And of course, they have the protein, right? Of course, they have the protein containing DNA, right? And that's in the head. And of course, this is how the structure looks, right? So we know that we don't consider them as living because outside the body, they'll not be able to do any process. Once they're inside the body only, they'll be able to do the process. Very good. I can see the correct answer in the chat. Right over here we have. Viruses are said to be acellular as they are not made up of cells. Right? Acellular. One cell is there. Not even one single cell, but they're just acellular. They have genetic material but is enclosed in a protein coat and not in the nucleus. They lack cell membrane or any other organelles. That's why we call them as non-living, and apart from that, they have the ability to reproduce only when they are inside the body. Yes, everyone? Yes, are we clear? Oh, guys, you are talking. Nice, Lakshmi. I think Lakshmi, Chitra and other people are having some serious conversation. Come on, focus in class. Very good. I can see lots of thumbs up. Very good. So over here we have it. And we are done with this question. Lots of thumbs up. Very good. Let's quickly look at the question number 11. What would happen if the endoplasmic reticulum is removed from the cell? Now, of course, what do you think? We know that ER is important, right? There are 2 ER. Smooth endoplasmic reticulum that actually helps in the formation of lipids. Then we have rough ER, which helps in the production of proteins. And we know that both of these are important, right? Over here, we have to mention about the membrane biogenesis, right? They actually help in the formation of the plasma membrane. Yes? Very good. No proteins, no lipids, no cell basically. So over here, we have this understanding about the ER, right? There's a network of the reticulum, channels for the transportation, synthesize the lipids, and of course its help in the membrane biogenesis. We are talking about the smooth ER. Then we have the rough ER, which of course have a ribosome that plays a very important role in the synthesis of proteins, right? So of course if these are not there, we know what will be happening. The endoplasmic reticulum is very important because it helps in the, synthesis of plasma membrane, proteins, steroids, and the lipids altogether. Furthermore, of course, if the absence of ER is there, the cells will not be able to synthesize the enzymes that are important. Then of course everything will be falling apart, right? So this is how you can frame your answer, everyone. Okay, Purnis, I'll explain you. So we know that ER is important. It's a very important organelle. ER actually is of two types in cells. When we talk about smooth endoplasmic reticulum, it does not have any ribosomes onto it, and that's why we call it as smooth ER. It helps in the synthesis of lipids, and lipids plays a very important role in the synthesis of the plasma membrane, and of course helps in other processes also. Then we have rough ER, which helps the synthesis of proteins, and we know that proteins are the building block of the cells and of the body. So if these are now not there, cells will not be able to do anything. So I hope that this is clear, right? Purni, I hope that you got the answer. No cell can exist without ER. Very strong statement. Yes, absolutely correct. Great. Lots of thumbs up, everyone. Awesome. Moving to the question number 12. Drums rolls, everyone. We are just at the end of the class. Need your support for some more time. Explain how do cells wall permit cells of fungi to withstand very dilute external medium without busting. Now I want to see how you will be responding to this question. It's for 3 marks, right? We're talking about fungi. Now, fungi also have a cell wall, right? But their cell wall is different, right? Cell wall of fungi. First, tell me the cell wall of fungi is made up of? Very good, chitin, right? Very good, Prachi. Very good. It is made up of the chitin, right? Yes. Now, of course, we know that we have the chitin, right? And of course, we have the cellulose, which makes the plant cell wall. Now, both of these are very rigid for sure, right? So, let's understand this further. So, of course, we know that fungi, cell wall is composed of the chitin, and it protects it from any of the worst condition. So, over here, we are taking an example of a plant cell. But don't worry about it. The only thing which has changed, of course, over here is that just consider that it's a fungi cell, and of course, we have it in the fungi cells, right? So, very similar to the plant cells, we have the fungi cells where, of course, even if the water is moving inside, it will not burst, right? Because it has the chitin, it has the cell wall. So, the turgidity is really very important over here. It will actually be able to take the pressure. So the pressure is there on equal side, inside, outside, and that's how it works. And that's why the cell will not bust. Very good. When the cell will expand, the cell wall pressurize the expanding of the cell and swells more, thus providing the turgidity. Very good, Rashmi. Very good, yes. Very good, Rukhaya. Yes. Chitin. So we are clear, right? This is how we can frame our answer. Here we have fungi possess a very rigid and sturdy cell wall made up of the chitin. Chitin is complex structure, right? Substance which provide the fungal cell wall from any of the adverse condition. When placed in a dilute solution, the water enters into the cell. Due to the endoosmosis, the cell will swells up. The cellular content will exert a pressure on the cell wall, right? The cell wall prevent the busting of the cell by exerting pressure back onto it, which is known as cell wall pressure. Are we clear? So on both the sides there's a pressure. So if we go back over here, over here, right? So if we have this, if there's a water moving inside, of course, there's a pressure, right? And of course the cell wall is also putting the pressure. Yes, Balram, please write your doubt. Please write your doubt. Mahalakshmi, ma'am, is every cell wall is made up of chitin? So Mahalakshmi, the cell wall of fungi is made up of chitin. The cell wall of plants is made up of cellulose. Right? Are we clear? Please, Balram, write your doubt. Write your doubt. Right? Are we clear? You can make a diagram also, right? So, of course, the other organelles that are there, right? The cell wall will be preventing it. Of course, we will have the cellular content, the content will be putting a pressure, and of course, the cell wall is also putting the pressure. Yes. Okay, I'm just bending a little bit over here to see your chat. I hope it's clear, right? Balram, I'm still waiting for your question. Turgidity means that it's very rigid. For example, there's an equal pressure from both the side and the cell is in there in a very swell up condition. No issues, no issues. Very good. Yes. Yes, Rashmi. Yeah, if mitochondria is not there, of course, we'll see no production of the energy. When the cell expands, the cell wall pressurizes the expanding cell to swell up even more. Yes, Balram. So over here, as we mentioned that, so pressure is happening, the water is moving inside, right? Of course, that is also a pressurizing situation. So the cell is swelling up. The content that we have will push the pressure on the cell wall, and cell wall will also push the pressure. So it will like equal like this. Nice. So with this, we are done with this question. I hope that Balram, I have cleared your doubt. We will be having the tissues chapter really very soon. So yeah, fun study, you can wait for that. Dilute means it has more of the water, right? In dilute condition, more of water. Endoosmosis means water will be moving inside the cell. That is endoosmosis. Very good, Himani, very good. Yes, Rukia will have tissues very soon. Nice, everyone. Okay, here we have question number 13. What is membrane biogenesis for 2 marks? Quickly, everyone. What is membrane biogenesis? We have just discussed. Irfan, endoosmosis means the water will be moving inside the cell. Right? Endoosmosis. Okay, very good. I can see someone was asking for the difference between the mitosis and meiosis. Mitosis is the equational division where the number of the chromosomes in the cells will remain same and it will give rise to 2 daughter cells. Meiosis is a reductional division where the number of chromosomes will get half and at the end will have 4 daughter cells. Very good, very good. Very good, yes, if the membrane biogenesis. We know that membrane biogenesis helps in the formation of the cell membrane. It has, of course, the proteins and the lipids all together. So, of course, this plays a very important role. We get the lipids and the proteins from the ER. So, that's why they're really important. So, you can frame that membrane biogenesis is the formation of or the renewal of the plasma membrane from its constitute, right? It's basically from its constitute, which is lipids and the proteins. And the lipids and proteins are synthesized by the smooth and the rough endoplasmic reticulum respectively. So you can, over here we have the definition of the membrane biogenesis, right? Yes, very good. So this is a very easy answer, right? For 2 marks. Okay, now I have a very difficult question for you everyone. Read this question. Explain what happens when a drop of concentrated solution is placed on a Rio leaf, basically on a leaf, and mounted on a glass slide. Name this phenomena. Would the same will happen if the Rio leaf was boiled before mounting? Give the reason for your answer. It is important. I'm sure you can easily tell about this, right? So we basically have a leaf. If we boil it, what will happen, right? If we boil the leaf, what will happen? Or if we have the leaf, not the leaf like this, yeah, leaf like this, right? And if you mount it on the microscope, what will happen? Very good, I can see the answer, right? Let's quickly take a look over here. So of course, in the living cell, what we'll see, we'll be able to see the things properly, right? Of course, it's living cell, of course, nothing will happen. We'll see the process of plasmolysis, right? Right, of course, right? The water will move, and of course the cellular content which is there will move in a one side, will actually move away from the cell wall, right, over here. So this is plasmolysis. Of course, in the boiled cell, we'll see nothing, right, because of course it's dead. So over here we have the answer. Very good. I thought it was a very trickier question, but you guys made it really very easy. So when a drop of sugar solution is added, what will happen? We will see the phenomena which is plasmolysis. Water will move out, right? So water will move out, right, and does not occur in the cell because it is boiled, therefore killing the cell, right? Exoosmosis will occur only in the living cells. Exoosmosis is nothing but the when the water molecules will move out. Endocytosis, Irfan, endocytosis is the process by which the organism will be engulfing something, moving inside. Endocytosis. We usually see this in amoeba. Are we clear? Yes. Everyone, come on, come on, come on. Are we clear? Very good, very good. Last question, everyone, last kind of, not last question, but yeah, second last question. Question number 40. A fruit is green when unripe, but become beautiful color when ripe. What does this change occur? Right? Of course, we have seen beautiful green color fruits, right, turning into beautiful colors, right? So what is the change that we have? Purni, I will explain. Bye-bye. Those of you who are leaving, just give me a minute. I just dropped my pen. Yeah. Yes. Very good. Yes. I can see the answers. Of course, we have the chromoplast, which actually is a color plastid, and because of that only we have beautiful colors in the fruits and in the flowers, right? And of course, it gives the bright color. And of course, we usually see that in the raw fruits, we have the chlorophyll. Of course, it gives the green color, but of course, as it becomes ripe, we'll have the beautiful bright color. So there's a very important reason for that. The green color of the unripe fruit is due to the presence of the chlorophyll. And as this start to ripe, this is important, everyone, the chlorophyll that is there in the chloroplast breaks down, right? And other pigments such as the carotenoids are synthesized by the plants in the chromoplast. And because of that, what happens? We will see the change in the color. So this is important that breakdown is happening, right? And as a breakdown happens, so carotenoids are synthesized by the chromoplast. Yes. Are we clear? That's nice, Himani. Very good, very good. So we got this question. Thank you, Rashmi. Irfan, Rashmi have explained you that. Next question, everyone. What is the importance of this change? Now, when there's a change from green to some bright color, what is the importance of it? Of course, we know that it's ripened and we can easily eat. Anything else apart from that? Just a 1 marks question. So we know that unripe fruits are green because of the chlorophyll. As they ripe, the chlorophyll breaks down. And of course, we'll have other colors, right? And this colorful chemical reaction in the ripening fruits helps is to preserve it. And of course, signal the animals that they are ready to eat. We also eat, right? And of course, to attract the pollinator. Very good point, Rashmi. Very good point. Very good, Karthikeyan. Yes. Yes, we can also eat, attracts animals, and we know that it's to preserve it also. Awesome. Now everyone, last question of our session, right? Question number 15, you have to describe the diagram of the fluid mosaic model of the plasma membrane. Now, it's a 4 marks question. So, in this particular question, if it comes in your examination, you need to draw this diagram. You can easily draw by, putting like this, a long membrane, right? On both the sides, phospholipids bilayer, right? You can show the proteins, you can show the carbohydrates right over here, and they plays a very important role. And of course, you can mention about the points. So, structure is important, and then of course, the way you will write your answer will depend. So, the structure of the plasma membrane was described with the help of the fluid mosaic model, which was suggested by the Singer and the Nicholson in 1972. It came in your examination. I was just telling them that these questions are important. So, yeah. Over here, we have it in 1972, right? So this is a very important question. Bye-bye, fun study. According to the plasma membrane, it is made up of the bilayer, which is of course of the lipids, phospholipid, right? Two layers are there. And these two layers, of course, we have proteins also which are floating in. Apart from that, you can mention that each phospholipid has a hydrophilic head, and the outside and on the inside we have hydrophobic tail, right? And of course, in between we have the bilayer, and we have cholesterol and the proteins which are embedded in the plasma membrane, and they just gives a look, right? And of course, each of it has a important function. Very good. Yes. So are we clear, everyone? So this is really very important question. I hope that you got this answer. It's okay. It's okay, Rashmi. You can easily draw this. Yes. I hope that Himani, you wrote your answer. You wrote the answer for this question. Awesome, everyone. So are we clear with this? Those of you who have joined a session in between, right? If you have any confusion, please come back, watch this video, because see, again, it's important that for you to focus on the questions and how you will be writing the answer of these questions in the examination. So I hope that you have enjoyed this session, and with this we are done. I'll just quickly take you through again to the mini learning program that we have. Do use the code YTFree for the amazing experience and amazing classes. You know that we have got you covered, and don't forget to hit the like button for this video, share with your friends and subscribe to the channel. Yes, everyone, I hope that you've enjoyed this class. Yes, thank you so much, everyone. Thank you so much. I can see the chat. Bye-bye, everyone. The means removal. Sorry. sorry, Ranjit sir. I can see one interesting question. Detoxification means nothing but the clearing of the toxic waste, right, from the cells. Yes. Right, in that also we have a very important organelle that plays an important role. Golgi bodies, right? Yes. Great, everyone. So now I'll say bye-bye to you. Take care of yourself, and we'll be meeting in the next class. If you have any doubts, any inquiries, please write your question in the comment section below. We'll be able to answer your questions. Bye-bye, everyone.